Hello guys, and welcome back to another episode of Last Minute a channel that helps you with your life sciences, whether it's the night before your exam and you're so stressed out about what you're going to be writing tomorrow, or you're just starting this new section in school and you think it's intimidating. This channel will make life easier for you. So today we are going to be looking at genetics. We're going to go through the punnet cross, how it's done, and all the other features that are involved in it, especially in genetics. We're going to be looking at sex-linked traits and the pedigree and how you use the pedigree, genetic modification, cloning, mutation, and the Mendel's laws. So let's get into it. So firstly, we're going to go through the monohybrid cross using the punnet square. So the monohybrid cross is going to be a cross that we're going to be looking at just one trait. Mono means one trait. So for example, it's either you pick eye color or height or hair and you do a cross based on that. And we're going to be using a punnet cross here, so you will see how we use a punnet square to see how traits are going to be passed down to children between mother and father. So we chose to do a cross between a trait which is going to be brown eyes, and as you can see here, mom has brown eyes and dad has blue eyes. So we're going to use the punnet square to determine which trait is going to be passed down to the children. So if you look at the mom's alleles, which is the B and the small b and the big B are alleles. Those are individual alleles. Since you know one allele has 23 chromosomes, and if they're two, that means it's 46 chromosomes. So as you can see, mom's alleles are heterozygous, meaning one allele is capital letter and another allele is small letters, or you can say one allele, which is the capital letter one, represents a dominant allele. And a dominant allele is the allele that will present itself physically. That's the trait that is going to be in the majority of the family members, and it's also going to be the trait that you will see. And as you can see, the other allele that mom has is called a recessive allele, which means mom carries the allele of having blue eyes, but she doesn't. Instead, she presents having brown eyes because her dominant allele for her brown eye allele is the dominant allele. That's why it is presenting. And then the recessive allele is the one that gets masked and only needs two recessive alleles for it to actually present physically, which is why dad has two small letter B's, which represent blue eyes, the trait blue eyes, and that's why dad presents physically having blue eyes. And since he has two the same recessive alleles, meaning they're both small letter, he has homozygous alleles. So he is homozygous. Homo means same and identical, and that's why his alleles, as you can see, are identical because they both are small letter, and mom's alleles are not identical, and that's why they are called heterozygous. Hetero means different. So because mom's alleles, one is a capital letter and the other is a small letter. So we have explained that. So now we are going to do the cross. So when obviously we look at these alleles, as you can say, we take them as when mom and dad have a child, 50% of mom's genetics are going to be involved and 50% of dad's genetics are going to be involved. This is how you do a punnet square. So you draw up like this, and then you separate. So we have mom's alleles, the big B and the small b is separated, and so is dad's. And then once you start binding them together, so you bind them like this. You look at the B at the top, you bind it with the first B, the second B with the second B. So that's how it is done. I hope it makes sense. So then you get your genotype. So genotype is your genetic makeup. So the genotypes that we have here, we have two different genotypes. We have two of big B and small b, which are heterozygous, and we have two homozygous genotypes, which are small b's. So that's our genotypes. It's the genetic makeup. Then a phenotype is what's gonna present physically. And as you can see, there's gonna be, let's say, 2 children that are gonna have brown eyes. As you can see, the 2 heterozygous alleles over there, they code for brown eyes because there's the big B and the small b together. And then the other ones, which are 2 small b's, represent blue eyes. So there'll be 2 kids that are going to have blue eyes. Let's just look at that. But then when you look at the ratio, basically what this means is that there's a 50% chance that some of the kids that they might have will have blue eyes, just like dad, and some of them might have brown eyes. So it's a 50-50 chance, or 50% chance that either the kid that they're going to have is going to have brown eyes or blue eyes, because this is a cross between heterozygous and homozygous. So I hope that makes sense. But yeah, you can play this over and over again, this section, to slow it down so that you can be able to understand. So now we're going to look at a dihybrid cross. A dihybrid cross is very complex. I'm gonna try to see if I can break it down as easily as possible for you guys to understand. So for dihybrid, we're gonna be looking at 2 traits. 2 traits. So for example, in this the example I'm gonna use here in this cross, we're gonna be looking at apples. So for example, the capital R is gonna code for round, and then the small r is gonna code for wrinkled. So here we have 2 alleles. One dominant one is gonna code for a round apple, and the other small recessive one is gonna code for a wrinkled apple. And another trait we're gonna be looking at is the color of the apples. So we have capital letter Y, which is going to be for yellow apples, and small letter y, which will be for green apples. So we're going to be looking at a cross to see, okay, so how many apples are we going to have that are going to be round and yellow, or round and wrinkled, or wrinkled and yellow, or wrinkled and green? See, so it's 2 traits that we're going to be looking at the same time. So here the cross is going to happen between a round and yellow apple with another round and yellow apple. Okay, so when you look at the alleles, you can see that because capital letter R is with small letter r, that means round is the one that's presenting because it's dominant. Then the small letter r, which is wrinkled and recessive. And then the second trait, you can see capital letter Y is the one dominant, and that's for yellow, and small letter is for green. So this apple is yellow. Okay, so now we're gonna do the cross. So here, what we do here is a bit different than the mono hybrid cross. So first, before you start drawing your squares, you first have to separate the traits and the traits. So this is how you're going to separate it. So you're going to look at the R, and this R, you're going to pair it with the other allele. So the alleles need to pair. So you're going to take R and you're going to pair it with the capital letter Y and also in small letter y. So you're going to write that down. So it's going to be capital letter Y and small letter y, or the first allele, which is different for the different traits, and the second allele of the different trait. Then you're going to do the same thing with the small letter r. You're going to say the second, you're going to take small letter r and you're going to bond it with the capital letter Y, and so on with the small letter y. So that's what you're going to do. You're going to do the same thing again for the other apple. You take the first allele of the first trait, you connect it with the first allele of the second trait, first allele of the first trait with the second allele of the second trait, and you do the same thing with the second allele of the first trait. Look at the video, and I've also, because you can see the arrows over there, I hope you can see what I'm trying to say. So then now we are getting ready to draw our squares. So we gotta write everything out spaced out, because during metaphase 1 or metaphase 2, the genes or the alleles, they get independently sorted and they get segregated and they independently sort without influencing each other to increase a variation among the family. So here we're looking at a family of apples. So now we're getting ready for to start our punnet square. So you have to separate them like this, write down every combination as is, and then once you're done, then you're gonna start bonding them. So when you start bonding them, you're gonna keep the alleles of the same trait together. So you're gonna say the R is gonna connect with this R, connect the R with the other R for the other apple, and then you write down what's gonna be the combination. So for here it's double R. You do the same thing for the second trait, you connect it with the same trait. Does that make sense? So when you're doing this bonding, you have to take the same traits have to be with each other. So the alleles that code for the same trait have to be together. And it's always best to write the one with the capital letter in front when you're doing this. For example, like if you have, so when we're doing the small R and the big R, notice that we write the big R first, then the small r, just so that you don't make a mistake when you're counting to find out, what traits does this apple have, so that you don't mistakenly say, this is a wrinkled apple, but because you looked at the first part and you didn't notice that there's the R in the middle, and because that R is big, it's dominant, so it's going to mask the recessive allele, which is the small letter. So make sure that you write it like that. And also just be careful, you make sure that you're writing everything correctly during your test, so that you also don't mistakenly write the wrong allele or the wrong trait. See, I already made a mistake myself here when I was writing. So we're just gonna keep doing that. So here is the final product of everything. So then now you're gonna have to count, because you can't write all of these genotypes down. As if you remember, guys, genotypes, the genetic makeup. What we were just writing here in this square, it is genetic makeup. So now we have written down the phenotypes instead. So you can count how many the genetic makeups that code for this particular thing. So we found out that here we have 9 round and yellow apples, 3 round and green apples, 3 wrinkled and yellow apples, and 1 wrinkled and green apple. And that was the final one over there. So this is how you do your dihybrid cross. When you do the cross, make sure that you take your time, slow down, think about it, make sure you wrote everything correctly, because they are really, really tricky, as you can see with this dihybrid cross. Now let's look at multiple alleles. In multiple alleles, it means that a gene has more than one or more than 2 alleles that code for it. For example, that codes for one specific trait. For example, blood types. Blood types have different alleles, but they all are for blood type, that trait. For example, instead of having a dominant R for red and a small or recessive R for pink in flower colors, for blood type you can either have the allele A or the allele B or the allele O. So you have 3 alleles and they are all for one trait, which is blood type. So we're going to be crossing a mom with blood type O and a dad with blood type A. B. So why, oh, I'm sorry, not O, blood type A. So the reason why mom has blood type A, because blood type O, or the allele for O is recessive. So that's why mom is considered to have blood type A, even though the alleles she has are the A allele and O allele, but the O allele is recessive. And dad has blood type A and B because because the alleles for A and B are both dominant. So what you will have here is both of them presenting. So we're gonna cross here the A's, as you can see, AO and AB and BO. How we're gonna do the cross. This is how you do the punnet cross for multiple alleles. And so now when we look at the genotypes, which is, again, if you remember, the genetic makeup, we have AB, AO, AB, AA, and BO. So the phenotypes here, we have blood type A, which is two of those, AA and AO, because O, the allele for O, is recessive. And another blood type we have is blood type AB. And the third blood type that we have here is blood type B, because again, the allele for O is recessive. The other alleles are dominant. Again, we have blood type A, which is represented by AA and AO, blood type B represented by AB, and blood type O, as I said, it is recessive, which means that it would need two alleles for it to present. So when a person has blood type O, they have both the alleles for O. For the trait blood type, there are 6 genotypes and 4 phenotypes. Phenotypes, we talked about this, that is how the physical presentation. So in the genotypes is the genetic makeup. So we have 6 genotypes in blood types. We have AA, AO, BB, BO, AB, and OO. But the phenotypes, or the type of blood types we have blood type A, blood type B, blood type AB, and blood type O. We only have 4, so that you can only physically show these. But the genetic makeups, there are 6 types or different types of genetic makeup for blood type. So I hope that makes sense. You can slow down the video, watch again. We are gonna be also looking at incomplete dominance. So incomplete dominance is when you have 2 traits that are both dominant and they end up just blending together. So the example we're going to use here today is a red flower crossing with the white flower. So the red gene or trait is dominant. The alleles are dominant. The white trait or allele is also dominant. But then so incomplete dominance, they just end up blending and they form a pink flower. So you can do the cross like this. You cross the R with a W, the R with a W, and then RW is not going to be red and white, but instead it is pink. So that's what happens. Again, genotype is the genetic makeup, which is RW, and then the phenotype is how it's going to show. And again, instead of saying it is red and white, it blends and forms pink. So the flower that's going to form is going to be pink. So that's what happens in incomplete dominance. And then in complete dominance, remember the first one we did in the monohybrid cross, the brown eyes or the brown allele, the dominant allele, was the one that was dominant. So complete dominance is when the dominant allele is the one that presents. Sex-linked traits are traits that are found on your X or Y chromosomes. Your X or Y chromosomes are your sex chromosomes, but most sex-linked traits are normally found on the X chromosome. And normally, if it's a trait that is found on the X chromosome, the male will normally have it. For example, for color blindness, color blindness is more prominent in males, and the reason being is that for females we have XX, but for males there's XY. So the allele for color blindness is recessive, so you need double of those for it to be present. And since for females, a female can be a carrier for color blindness, but if she has another X chromosome and that chromosome has a dominant allele, then it masks the color blindness. But yet for a male, if he has this recessive allele, even if it's just one recessive allele, he still presents or shows signs of having color blindness, because there's no another X way that can mask this. He only has Y. I hope it makes sense. So color blindness and hemophilia, which is when you don't have blood clotting factors in your body, so you just excessively bleed and you don't stop bleeding. That's another disease that is an X-linked disorder, and it's found on the X chromosomes. So you can look at the punnet square over there. You have the female, which has the recessive alleles for color blindness. This female has color blindness because she has both the alleles. And then the male who doesn't have color blindness. And then when you see the X, the daughters are both carriers. Their mom had color blindness, but the daughters do not have color blindness. Then when you look at the sons have color blindness, yet their father does not have color blindness. And again, just like I mentioned, it's because they don't have another X chromosome that's gonna mask it. So that's how that works. And then we're gonna look at the pedigree. The pedigree is this diagram that I have there, and it basically shows how specific traits or genetic conditions are inherited through generations of a family. So it's like you're looking at the family tree, but looking at the traits. You can even help, I wouldn't say it helps you see who's related to who, but it just, in the family tree, you can be able to track a certain genetic disorder. That's why if a woman is pregnant, or before you even get pregnant, if maybe you're getting married, you can go with your husband or soon to be husband to the doctor to get a genetic testing. So they'll look at his genes and they look at your genes, and maybe you can find out if any of you are carriers of any genetic disorder, and in that way you'll be able to be much more informed when you're about to have a baby, decide to have a baby. Or sometimes even if you may not know your medical history and you didn't do that prior to getting pregnant, but once you get pregnant, you are given the choice to find out what's going on with your genetics, so that you can know your child's genetics, mostly not even yours, your child's genetics, because you already have the child inside your womb. You are able to check to see if there's any signs or any alleles that's coding for specific genetic disorders, in order for you to prep for the entire pregnancy. You'll be able to prep for your baby. So if maybe your baby is going to come up with a genetic condition, the doctors can able to prep accordingly for the delivery, and you as a parent can also prep how you're going to be able to make life easier for your child if they have a genetic disorder. So that's the importance of having these pedigrees. And as you can see, the square is represents the males. This is how it always is. The square is for males and the circle is for female. And if these shapes are colored in, that means you have the disease or you're a carrier. As